Hello everyone. Welcome back to our plant day. In today's lecture, we'll learn all about the life cycle of angiosperms, part 2, in a simple and easy to understand way. So, let's begin the life cycle of angiosperms, part 2, forming the wall of the anther. The outermost parietal layer, lying next to epidermis, constitutes the endothecium. The cells of endothecium develop fibrous bands of thickenings and assist in the dehiscence of anther on maturity, at a point called stomium, where the endothecium is thin-walled. The innermost layer of parietal tissue develops into a single-layered tapetum. The cells of the tapetal layer are radially elongated, wedge-shaped, and have dense cytoplasm and prominent nuclei. In between the endothecium and tapetum, 1 to 3 layers of cells are present. The cells of primary sporogenous layer divide mitotically and give rise to microspore mother cells or pollen mother cells. In the beginning, the microspore mother cells are closely packed, but as the anther enlarges in size, the pollen sacs also increase in size. These cells enlarge in size, become spherical in shape, and get loosely arranged. In many cases, some of the spore mother cells are non-functional and act as food for the rest of the cells. Various stages showing development of microsporophyll and microsporangia. Microsporogenesis. Each microspore mother cell produces 4 microspores or pollen grains. The nucleus of each spore mother cell divides twice to form 4 nuclei, the first division being reductional, meiosis I, and the second being the ordinary mitotic one, meiosis II. The nuclei are haploid. The original wall of the microspore mother cell becomes mucilaginous during the formation of the microspore, and a new wall is formed around each newly formed pollen grain. In most of the dicots, the wall formation is simultaneous type, i.e., the nucleus of the spore mother cell divides twice, forming 4 haploid nuclei, and the walls are laid down between the 4 nuclei simultaneously by furrowing method. The 4 daughter cells formed are arranged in a tetrahedral manner, forming a tetrad. In majority of monocots, the wall formation is of successive type, i.e., the cell wall is laid down between the 2 daughter nuclei immediately after their formation during the first meiotic division. After second meiotic division, again a wall is formed in each of the 2 daughter cells. This results in the formation of a tetrad of cells. The tetrad formed is termed as isobilateral tetrad. In still other cases, the spore mother cell may divide transversely, giving rise to a linear tetrad as in many Asclepias. In some cases, 2 spores may have linear arrangement, while the other 2 may have a transverse arrangement, resulting in the production of a T-shaped tetrad. Example, in Aristolaceae. In Magnolia, inverted T-shaped or decussate tetrad is found. Different types of tetrads. A. Isobilateral. B. Linear. C. T-shaped. D. Inverted T-shaped stroke. Usually pollen grains separate from one another at maturity, but in some cases, as in Typha, Drosera, Juncus, the 4 cells form a group and do not separate. In Calotropis and orchids, the pollen grains in a pollen sac unite into a mass, forming a pollinium. A pollinium, when the pollen grains mature, they exert pressure from within on the wall of the anther. The anther bursts and sets free the pollen grains. The dehiscence of the anther takes place in the following ways. 1. Longitudinal. The anther lobes burst in longitudinal direction, e.g., in Datura, Helianthus anus, sunflower, hibiscus, gossypium. 2. Transverse. Anther lobes dehisce breadthwise as in Ocimum. 3. Porous. The dehiscence take place through pores, e.g. in Solanum species. 4. Valvular. The dehiscence take place by 1 or more valves, like the shutter of a window, which open on the outer side only, as in Berberis. Types of dehiscence of an anther. A. Longitudinal. B. Transverse. C. Porous. D. Valvular. Dehiscence of microsporangium. When the anther is mature, the middle layers and the tapetum disorganize. The sporangial wall now consists of epidermis and endothecium. The sterile partition between the two pollen sacs disintegrates, and two pollen sacs of one side unite together, forming a single compartment. The pollen grains are released through stomium, generally strong microspores. Each microspore is a unicellular, unenucleate structure, possessing two coats, the outer exine and the inner intine, and contains a large central nucleus and dense cytoplasm. The exine is thick, cuticularized, and tough layer, and it is often provided with spinous outgrowths, warts, or reticulations, and sometimes smooth. The intine is thin, delicate, cellulosic layer, lying next to exine. The exine possesses one or more thin places called germ pores, through which the pollen tube grows. A microspore germinates on stigma and form a long tube, known as pollen tube, which traverses the style and reach embryo sac. The intine is thin, delicate, cellulosic layer lying next to exine. Mature pollen shows the following composition: proteins 7 to 26%, fats 1 to 14%, carbohydrates 24 to 48%, ash 1 to 5%, water 7 to 16%. Male gametophyte. The microspore pollen grain is haploid. Germination of the male gametophyte starts while it is still in the microspore nucleus, is shifted to periphery due to the development of a central vacuole. First division results in the formation of a generative nucleus and a vegetative nucleus. The generative nucleus, along with some of the cytoplasm, is delimited from the rest of the cytoplasm by the formation of a membrane and forms the generative cell, while the remaining part is the vegetative or tube cell. The nuclei of the generative and vegetative cells differ in size, structure, and in staining qualities. The nucleus of the vegetative cell is with a prominent nucleolus. The cytoplasm is rich in RNA and poor in DNA. Starch and fats are the conspicuous substances present as food. On the other hand, the nucleus of the generative cell has a small nucleolus. The cytoplasm is poor in RNA, less stained, rich in DNA, and poor in proteins. Finally, the generative cell loses its contact with the spore wall and is shifted into the vegetative cell, where it may lie in any part of it. The generative cell is generally biconvex, but in some cases, it is elongated. Generally, the microspores are shed from the microsporangium in two-celled stage. The generative cell divides to produce two male gametes. In majority of the cases, the generative cell divides after pollination. Pollination, the transfer of microspore, pollen grains from the anther to the stigma, is called pollination. The pollination may be of two types. 1. Self-pollination, the transfer of microspores, pollens, from the anther of a flower to the stigma of the same flower. 2. Cross-pollination, the transfer of the microspores, pollens, from the anther of a flower to the. If you found this video helpful, please give it a thumbs up, share it with your friends, and don't forget to subscribe for more easy biology lectures. Also, add 3 questions at the end of lecture. Thanks for watching, and see you in the next class. Bye.